Hello students, now we will discuss about new chapter cell cycle, cell division. In this chapter, we will discuss about mitosis, meiosis and amitosis and different types of division enhancing substances and division stopping substances also. In each and every cell, when it has to undergo cell division to increase its number or increase in mass or to produce the next generations, it first of all will undergo some preparatory stages or preparatory phases. So that preparatory phase in case of cell division means before undergoing cell division, whatever the steps that the cell is undergoing for preparing itself for cell division is called as interphase. So we will start with few important points. Cells divide by few methods like mitosis or meiosis or amitosis. And before undergoing cell division, any type of cell division, it can be either mitosis or meiosis or amitosis. Every cell will undergo a preparatory stage, means it will prepare itself for the division. Called as interphase. Previously, this interphase is considered as resting phase, means nothing changes will be happening inside the cell during interphase. Only some changes will be happening during the division time. But later it was observed that interphase is not a resting stage, but many changes are happening inside the cell just before cell division. So this is also called preparatory phase. During this time, cell is active and has many internal changes. There are many steps during interphase. We will see what are those. The steps are included or involved during interphase are: first one, G1 phase. Second one, S phase. Third one, G2 phase. G1 phase is called as gap 1 phase. S phase is called as synthetic phase or during synthesis phase. Here G2 phase is called as again gap 2 phase. So there are two gap phases and one synthetic phase. During cell division, so here we have written mitosis and meiosis. Cell division or cell dividing time or phase is said as M phase. M phase can be a mitosis phase or meiosis phase. Can be mitosis or meiosis. So during M phase, here we have written the steps included in interphase are G1, S and G2. In the same way, in M phase, what are the steps that are included? Here, steps included are First one, karyokinesis. When cell is ready for division, then first change happens inside the nucleus. Karyo means nucleus. Second one, cytokinesis. Cyto means cytoplasm. So cytoplasmic division and nuclear division is happening during M phase. Again, during karyokinesis, what are the steps included? Here, steps are 1. Prophase, second, metaphase, third one, anaphase, fourth one, telophase. In this prophase, metaphase, anaphase, telophase, these four are included both in M phase and both in mitosis and in meiosis also. Now let's start with interphase. Already we said that interphase includes G1 phase, S phase, third one, G2 phase. So we will say about each and every phase or step one by one. Coming to G1 phase. G1 phase is also called as gap phase. We can also use a term like growth phase. Here what is happening? Whatever the raw material that is necessary for S phase, means whatever the requirements are there for S phase, are synthesized here. Here, all raw materials required for S phase are synthesized. Next, means in G1 phase, cell gets ready for S phase. So, think that this is the time. This is not the circle. Think that just this is the time that is required for completion of interphase. So we will represent some time taken by G1 phase. Here I am writing this total circle is the time. Next, what here we are telling is getting ready means something is being synthesized in the G1 phase. What are synthesized in G1 phase? Here in G1 phase, proteins, enzymes, and enzymes are required for DNA replication. What is meant by DNA replication? DNA replication means during this process, DNA replication process, other copy of same type of DNA is synthesized. Here in G1 phase, proteins, enzymes required for DNA replication and all three types of RNA are synthesized. ATP is synthesized. And due to synthesis of all these biomolecules, size of cell increases. When size of the cell is increased means obviously means simultaneously at the same time cell growth might have taken place. So here in G1 phase, what we have said, here proteins are synthesized, enzymes are synthesized, ATP is synthesized, all enzymes that are required for synthesis of RNA, DNA, etc. And ATP is also synthesized here. Why ATP is synthesized? Because for synthesis or for any type of metabolic activity, ATP is required. Here we will see one important point. There are many steps previously we have written, that is prophase, metaphase, anaphase, telophase. During this anaphase time, if this is a cell and here aligned chromosomes have to be moved to opposite poles, this pole and this pole. For suppose this has to move this side, this has to move this side. For movement of one chromosome, nearly a cell needs 30 ATP molecules. Means the requirement of ATP is much more during cell division. So that ATP is also having very important priority during interphase and also that too during G1 phase. Next coming to S phase. There are some types of checkpoints here. Means every cell checks how much enzymes are required, how much ATP is required, how much proteins are required during gap 1 phase or gap 2 phase. Every cell will check how much is required and whether the formed biomolecules are correctly framed out or not. If there are any correction, again automatically cell will repair itself. It will be having self-repair mechanism. Now during S phase, what is happening? Immediately after G1 phase, the next priority goes to S phase. So S phase is called synthesis phase or synthetic phase. Synthesis phase. Here what happens, as we have said before, DNA replication. Here DNA replication occurs. Means other copy of same DNA. This is also termed as autocatalysis. That we will learn later. So here DNA replication happens. And also we said here all three types of RNA are synthesized in G1 phase. Why RNA are required? RNA are required because here some of the special proteins are synthesized called as histone proteins. For that protein synthesis, the step included is translation. For that translation, mRNA, rRNA, tRNA are required. So all these three are required synthesized here and the next protein synthesis happens here. So histone proteins are synthesized. For a protein synthesis, three types of RNA are required. So those are synthesized here. Later, by the process of translation, proteins are synthesized here in S phase. Centrioles are synthesized. Centrioles are some of the structures which are normally present outside the nuclear membrane, but during the cell division, the nuclear membrane will be disappeared and these centrioles will be forming special types of proteins called as spindle proteins or spindle fibers. Those spindle fibers will drag the chromosomes towards the opposite poles. That we will learn later, but here just remember that one of the cell organelle called as centrioles are synthesized, which forms spindle fibers that are necessary during dragging of chromosomes to opposite poles. In lower eukaryotes, 50% of histone proteins are synthesized in G1 phase and 50% of histone proteins are synthesized in S phase. But in higher eukaryotes, you can consider these important sentences as statements, which can be an MCQ. 100% of histone proteins are synthesized in S phase. So, the amount or quantity of DNA is doubled, but the number of chromosomes remains same. Whatever the total DNA content is synthesized by the time of S phase, that is given with a symbol called as capital C. So, the total DNA content in S phase is indicated as C value. What does it mean? For suppose, example, as we have said that quantity of DNA doubles but the number of chromosomes remains same. In a diploid cell during S phase, what might be the means C value, or what might be the content of DNA, or what are the numbers of chromosomes. As we have said that it is nothing but diploid cell. It means diploid we represent with 2n. Here we are telling C value means total content of DNA. So we can write 2C is equal to, during S phase there is no increase in 2n. Here n indicates number of chromosomes. Number of chromosomes. We are telling in a diploid cell during S phase. So, by the completion of S phase, what will be the value of DNA and also chromosomes? We are telling 2C, or the total content of DNA which is present in prior at the beginning of S phase, will be 2C. So by the completion of S phase, DNA will be doubled than chromosomes. So DNA will be 4C, because we are telling DNA replication means formation of other copy of that. Initially if its number is 1, by the completion of S phase it will be 2. So already we have written here it is C. So it will be 4C. 4C means the total DNA content by the completion of S phase has been doubled. Then what about the chromosome number? As it is a diploid cell, and we have said that number of chromosomes will not change. It remains the same. So it will be 2n only. C indicates content of DNA and n indicates number of chromosomes. So these are few points regarding S phase. Here we will learn about other important point also regarding G1 phase. Example of protein synthesized are CDK. What is full form of CDK? CDK means cyclin dependent kinase. This protein is necessary to initiate the cell division. Initiation means here this protein is very much important for transcription and translation process. CDK is necessary to initiate cell division. These are some of the important points regarding G1 phase and S phase. We will write here, as we have written here, some of the proteins are synthesized, enzymes are synthesized, ATP is synthesized. Here, DNA is doubled. Next, chromosomes remain same. There is no doubling. The number of chromosomes will be same. Here, centrioles are synthesized. Histones are synthesized. G2 phase or gap 2 phase. So what are changes happening during this gap 2 phase? During gap 2 phase, cell is getting ready for M phase, because the next stage will be M phase only. Now, here some proteins are synthesized, enzymes are synthesized, ATP is synthesized. But by the time of S phase, DNA has got doubled. Means some ATP might have been utilized here. Chromosome numbers will be remaining same. New proteins have been synthesized. Again, some of the ATP might have been utilized during S phase. Means when the enzymes, the ATP are being consumed, there will be some scarcity here. So whatever the decreased number of enzymes or required proteins or ATP are decreased here by the time of S phase, that will be again resynthesized and completes its synthesis by the time of G2 phase. So whatever enzymes and ATP that are utilized in S phase are resynthesized in G2 phase. Now, so here the proteins formed are, in addition to some of the proteins that are formed here, what are new proteins formed here? Tubulin protein. Tubulin protein is one that is formed here, and plasma proteins that are again synthesized in G2 phase. Tubulin protein. Tubulin protein is required for synthesis of spindle fibers that separates chromosomes during anaphase stage, and plasma proteins that work as carriers or transporters during the transportation of gases or any of the molecules from outside the cell to inside or inside the cell to outside. Here plasma proteins are of two types, extrinsic proteins and intrinsic proteins are of two types. One is extrinsic proteins, second one intrinsic proteins. These are two types. Again, the ATP is whatever synthesized is here might have been utilized for all these molecular synthesis. So we will write ATP is also synthesized again. Now, three types of RNAs are also synthesized. Here, we will write that point. During separation of chromosomes, anaphase. Two opposite poles of nucleus for one chromosome, 30 ATP energy is required. So, more ATP are synthesized in G1 and G2 phases. Apart from these types of G1, S phase, G2, there is other type of phase called as G0 phase. So, G0 phase. G0 phase. This is not go, this is G0. I'm writing it this. So, after M phase. After M phase means after mitosis or meiosis. Meiosis. If there is no need of a further division, then the formed baby cells after healing, they will enter into G1 phase. From G1 phase, if there is no need of again DNA replication, again following the total cycle again, then cells will undergo differentiation. Means, for suppose, think that here parenchyma cells are formed into one parenchyma cell has given rise to two baby cells. If there is no requirement of further cycling and again producing more number of parenchyma cells, then here itself those cells will undergo differentiation. Means they will be converted into other types of cells. That phase or that time is called as G0 phase. So those cells from here, they will enter into G1 phase. From G1 phase, they enter into G0 phase. If there is no need for further cycling, then baby cells will grow in size and undergo differentiation into new type of cells So entering into G0 phase is happening from which phase? Means after G1 phase, at the end of G1 phase. So we'll write that point also, which is very, very important. G0 phase happens at the end of G1 phase for differentiation. If the question has come, what is the phase that is giving rise to G0? Means after G1 phase or during half of G1 phase, the cell can move into G0 phase or resting phase, where the cells will get differentiated, but they are not undergoing further cycling process. So we are telling cell is entering into inactive stage, also said as G0 phase is inactive phase. Again, we have to remember that G0 is not undergoing or entering after M phase. After G1, the cell will enter into G0 phase or inactive phase. This process of entering into G0 phase, then it is called as differentiation. Specialized term is given, that is called as differentiation.